Welcome to section 21.6a. Okay, gentle people, what we're going to talk about in this lecture is we're going to delve into protein chemistry. Now, the reason we're doing that are proteins are considered natural polymers, and so right off the polymer chapter, we're going to talk about how biology makes polymers. Now, proteins can be classified in a couple of different categories: catalyst, transport, and information transport. A protein that acts like a catalyst is called an enzyme. Just like non-biological catalysts, enzymes in your body speed up biological reactions. For example, you have an enzyme in your body called alcohol dehydrogenase. What this does is if you are to consume an alcoholic beverage, you need to metabolize that alcohol. This enzyme helps metabolize it. If you produce a lot of alcohol dehydrogenase, this is the type of person that can drink a whole bunch of alcohol and not feel drunk. If you're one of those people that are one and you're done, well, that means you're not producing a lot of alcohol dehydrogenase. Another type of protein class is transport. Now, we discussed this before. One example is hemoglobin. Hemoglobin takes oxygen from places where there's a lot of oxygen, such as your lungs, to places where there's not a lot of oxygen, such as the end of your capillaries. And then lastly, we have proteins that control the information in your body. For example, if you need to utilize some of your fat storage, if you need to crank up the sugar levels in your body or decrease the sugar levels in your body, these proteins control that function and they deliver information to the rest of your body so that you can produce what you need to when you need to. Now, what we're going to be talking about in this section are protein structures. And there are three main categories of protein structures: fibrous, globular, and integral membrane. Now, we're going to talk about just the first two, and I just want to give you the basic background on these. So, fibrous proteins are proteins which are long and stringy. So they tend to be proteins where the chain or the polymer chain happens to be one long continuous chain, and it usually is going to lie flat. Now, most of these are used as structural proteins. So things like your muscles, your ligaments, your bones, your fingernails, all of these are made out of fibrous proteins. Now, what I should note is that 50% of the dry weight in your body are proteins. These fibrous proteins make up a considerable amount of your biomass. What your body exploits is the physical properties of these proteins, i.e., if they're stretchy, if they give you rigidity, if they give you flexibility, all of those things. They're not usually involved in any kind of reactions. They are just there for structure. On the other hand, we can have globular proteins. Now, globular proteins have their protein all in a bunch and kind of are like this mixed blob. So what I want you guys to think about is take a shoestring and then crumple up that shoestring and kind of roll it into a ball in your hand. And that's kind of the structure of a globular protein. Now, these proteins aren't a glob because they tend to be proteins that are solubilized in water. And so they are floating around, and being a glob makes them more soluble. And what is also important is these proteins tend to be distributed through your body. And so one of the biggest functions is globular proteins, or a lot of globular proteins are enzymes. And so what they're going to do is they're going to be involved in reactions and speeding up bioreactions inside of living organisms. Also, globular proteins are going to be those information proteins. They're going to go ahead and regulate things in your body, measure things in your body, and turn on and off mechanisms in your body. And lastly, they are our transport proteins. If you guys remember what hemoglobin looked like, you can have remembered how that protein chain was coming double backed on each other and kind of crumpled into a ball. So let's go ahead and talk about how proteins are made. And what I'm going to use to make proteins are your amino acids. So let's go ahead and break that down. We know the amine functional group is one where I have a nitrogen single bound to a carbon and then the rest of my molecule. So here's the general structure of an amino acid, and right here you guys can see the amine group. Now looking at the amino acid's name, you hear the word acid, and we already talked about one type of acid. That was the carboxylic acid. And you guys can see the carboxylic acid group on the amino acid structure. Now the amine and the carboxylic acid are connected by carbon. That carbon has one hydrogen attached to it, and it also has what we call the R group, or the variable group. Now I know this is going to be kind of tricky. So in organic chemistry, we said the R group was the rest of the molecule. It's kind of the unimportant thing. For some reason, biochemists still use the R, and this time the R is the most important thing in my molecule. The R group is the thing that changes, and this is what's going to identify what type of amino acid we have. So I want you to make note of that. In this case, R is the variable group, and it is how we define our amino acids. Now, some other things to know. Like I mentioned to you guys before, amines are considered basic. So I have a base on my amino acid. And I also have my carboxylic acid, which, as its name implies, is an acid. Now, it turns out when you put the amino acid under biological pHs, what's going to happen is I'm going to deprotonate this acid, and I'm going to move that acidic hydrogen to my base. So my acid is going to lose the H+, my base is going to gain that H+. And so what I get is my amino acid in this form. Now you'll see the amino acids in both of these where I don't deprotonate the carboxylic acid or protonate my amine. If it's in this form right here, where the hydrogen is on the amine and my carboxylic acid is deprotonated, we call this the zwitterionic form. A zwitterion is something that has a positive charge and a negative charge on the same molecule. So just be careful. You can see the amino acid represented in the zwitterionic and the non-zwitterionic form. So let's talk about how proteins are going to be made. Now, proteins are polymers, and what I'm going to do is I'm going to take my amino acids, and my amino acids are going to be the monomers to my polymer protein chain. So what I'm going to do is I'm going to do a condensation reaction. What you guys will see is that we have a carboxylic acid and we have an amine. And like I mentioned before, we can condense these two things. What I can do is I can lose an OH off my carboxylic acid, an H from my amine, and I'm going to make a unit of H2O. That means the rest of my amino acids are going to come and link to each other. What I'm going to do is I'm going to have that carbonyl carbon attached directly to that amine nitrogen. And so I'm going to form this bond right here. This right here is called the amide bond in the red box. Now, what you guys will notice is after I do this condensation, I still have an amine, I still have a carboxylic acid. So what I can do is I can go ahead and add more amino acids on here. So I can either put amino acids on this side and grow my chain this way, or I can put amino acids on this side and grow my polymer chain this way. So in this way, I'm just sticking a whole bunch of amino acids together via condensation. So let's talk about the amino acids that I can put together. Here on this slide is the 20 common amino acids. Now don't worry, I am not going to require you guys to memorize this chart. I will give you the structure of the amino acids. Now what you will probably do in biochemistry is you will have to probably memorize their name, and each amino acid is given a 3-letter abbreviation, and there is also a single letter abbreviation. I will give you the name and abbreviations for all the amino acids that you use in Chem 1C. However, what I want you to be able to do is look at the structure of that amino acid and tell me how we classify that amino acid. Amino acids are classified based on their R group. They can either be considered nonpolar, polar, or electrically charged amino acids. And what I want you guys to do is you guys are going to disregard all the rest of the amino acid and you're going to focus on the R group to make that distinction. So let's go ahead and take a look and really delve into this. So of the 20 common amino acids, 9 are considered to be nonpolar. And so here's what I mean by nonpolar. So here's my amino acid, and this right here that's highlighted is going to be my R group. Now what you want to look at is what is the R group made out of. So if we take a look at this list, H, CH3s, CH3s, C's and H's, C's and H's, C's and H's, and C's and H's. So if your side chain is only made out of carbons and hydrogens, then you consider that a nonpolar amino acid. Now I'm going to warn you, there are two that are kind of tricky, and that is methionine and tryptophan. What you will notice is that these have other things besides carbons and hydrogens. Methionine has a sulfur, and then tryptophan has a nitrogen in it. However, what you will note is that it is buried inside the R group. So even though it has those different atoms in there, they're kind of tucked in all these CHs, and thus they are considered nonpolar. Now, if we look at the other 11 amino acids, these are either grouped as polar or electrically charged. Now, there are 6 of them that are considered polar. What you guys will notice is they're not only made out of C's and H's. They have OH groups attached to them, they have SH groups attached to them, and then they have NH2s attached to them. So the top ones are considered polar, and what you will notice is all those atoms that are O's, S's, and N's, well, they're at the end of my R chain. And so that means the polarness is exposed to the rest of its environment. Now, the electrically charged amino acids are real easy to tell because what you will see is at the end of the molecule, you will have a charged group, meaning we have something that has a minus or a plus attached to it. So the long story short is if you have anything besides carbon and hydrogen, chances are it's either going to be polar or electrically charged, with 2 exceptions being methionine and tryptophan, where that other atom is deeply buried inside of carbons and hydrogens. All right, now that we've seen all our amino acids, we know that we can polymerize them via condensation. Let's talk about the hierarchy in describing a protein structure. Now, there is something called the primary structure of a protein. The primary structure of the protein is going to be the amino acid sequence, meaning how long is the chain of your amino acids? Also, what are the amino acids you're using and in what order do they appear? Now, if we take a look at this, I took an alanine, a glycine, and a phenylalanine. These are 3 amino acids. I condensed them to form the amide bond. And what I made out of this is something called a tripeptide. Now, a tripeptide, peptide means protein, tri means I made it out of 3 amino acids. So if I use 2 amino acids, we call that a dipeptide, 4 amino acids, tetrapeptide. Now, what you have when you create this tripeptide is you have them in a certain order. What I told you in this one is I put the alanine first, the glycine, and the phenylalanine. What you will note is the alanine is in what we call the N-terminus. The alanine has the amine still attached to it. Now, the phenylalanine is going to be on what's called the C-terminus. The C-terminus is going to be the one with the carboxylic acid group. These two things define the ends of my protein. Now, what you guys will see is that I can make a sequence of amino acids however long I want. In here, we go ahead and have a sequence that is 28 amino acids long. What you guys will see is that the number and order determine what that protein does. For example, your book gives you a very classic example. There's an amino acid sequence that is 9 amino acids long. One of these sequences is the one for oxytocin. Oxytocin is the stuff that is involved when a woman is pregnant for uterus contraction and milk secretion. If I take that same protein sequence and I only change 2 of those amino acids, what I will form is a different protein. What I will form is vasopressin. And vasopressin is something that is in every single human being. It's the stuff that controls your blood pressure and controls some of your kidney function. So just by changing 2 things, I change the functionality of my protein. With that said, let's go ahead and try to explore this idea. I want a tripeptide, and I want it to contain glycine, alanine, and serine. I want you guys to use each one of these amino acids once. How many different proteins or how many different peptides can I make using these 3 amino acids? All right, ladies and gentlemen, let's go ahead and tackle this one out. Now, I'm going to use abbreviations. I'm going to abbreviate glycine as GLY, alanine as ALA, and serine as SER. Now what I can do is I can form a protein with glycine first, alanine second, and then serine on my C terminus. However, if I were to go ahead and switch the order of that serine and alanine, I would get a completely different protein that functions differently. Now remember, I can keep doing this. So in this case, I can start out with the alanine, and then I can go ahead and put the other 2 amino acids on there, and then I can switch their order. And then lastly, I can have serine start off my amino acid sequence, followed by alanine, then glycine, and like the last ones I showed you, I can switch the order of the last 2. So what you guys get is that you can form 6 different proteins with these 3 amino acids. You guys can note that the number of amino acids you have, you can go ahead and take the factorial of that, and that will tell you how many proteins you can make. So for example, I had 3 amino acids that I have, so 3 factorial, and to remind you guys, 3 factorial means 3 times 2 times 1, which equals 6. If I had 4 different amino acids and I wanted to make a tetrapeptide and I wanted to use each one of them once, that would be 4 times 3 times 2 times 1, which would give you 24 different types of proteins. Now, what you guys will note out of this is the longer I can make my chain, the more different types of proteins I can make. And so if I had something that was 28 amino acids long, well, you can see that I can make a whole bunch of different types of proteins. All right, ladies and gentlemen, let's go ahead and talk about the secondary structure of proteins. Now, the secondary structure of proteins are going to be the short-range structure. These result in 2 major structures that we can identify, and that is called the alpha helix and the beta sheet or the pleated sheet. Now, the way this is formed is through the hydrogen bonding of a carbonyl carbon and an NH group. So let's go ahead and take a look at what I mean by this. All right, ladies and gentlemen, let's say I go ahead and I string together a whole bunch of amino acids. And right now, each one of these blocks represents an amino acid, and I'm doing the condensation over and over and over again. So what I'm going to do is I'm going to try to focus on 2 amino acids, and let's say that they're about 4 amino acids apart. So this one and this one right here. So what I can say is I have that first amino acid, a couple more amino acids, and then finally I get to that other amino acid that I want you guys to focus in on. And so I can go ahead and draw out that second amino acid, put the rest of the chain on there. And so here's what I want you to see. What you'll see here is that this right here is a very electronegative atom, so this is an H bond acceptor. And now what you'll notice is that this nitrogen right here, well, that's super electronegative and it contains a hydrogen, so this is considered an H bond donor. Now what you'll notice, we have a donor and acceptor of a hydrogen bond. So what I can do is I can form a hydrogen bond between these two things. Now, for these two things to see each other, what's going to happen is my protein chain is going to have to curl or spiral or corkscrew. Once it goes ahead and does that, then I can form this hydrogen bond. So the hydrogen bond is between the amide's hydrogen and then the carbonyl of a different amide. And you guys can see that corkscrew structure. Right here, I have my nitrogen. This is my hydrogen that can be involved in hydrogen bonding, and it is hydrogen bonding to this oxygen here in red. And you can see that this happens over and over again, where you have a hydrogen and an oxygen involved in a hydrogen bond. Now, to have that happen, my protein kind of spirals together, and this is what leads to the secondary structure of an alpha helix. Now, there's another secondary structure that I can get out of this. All right, gentle people, for the beta sheet, we're going to talk about the same hydrogen bond. We're still going to look at that hydrogen on that nitrogen, and we're also going to look at that carbonyl carbon. Now, instead of my amino acids being 4 or 5 apart, what's going to happen is I'm going to have my amino acid chain, and it's just going to go ahead and double back on itself. So the chain goes one way, it turns and goes along, following alongside the original chain. And so what can occur is I can get hydrogen bonding between that nitrogen and oxygen in the same way. Now this is going to lead to that pleated sheet. So let's go ahead and take a look at that slide again. And you guys can see it in this picture again. Here's my donor hydrogen. Here's my acceptor oxygen, donor hydrogen, acceptor hydrogen. And in this case, if my protein double backs on itself, instead of getting a curl-like structure, what I get is kind of this hairpin structure where the protein kind of lies flat on itself, creating a sheet of amino acids. This is what we call a beta sheet or a pleated sheet. Now, if you guys walk in the chemistry building, you probably have seen these structures laid out in the three-dimensional models of proteins. Whenever you see these corkscrew-like structures, well, that's them trying to tell you that it has an alpha helix there. If you see these arrows pointed in 2 different directions right here, this is trying to tell you you have a beta sheet involved in your structure. With that said, let's go ahead and talk about the tertiary structure. While the secondary structure told you about the short range arrangement, the tertiary structure is telling you the long range structure. In other words, it is telling you how the alpha helixes and the beta sheets are going to be arranged. For example, does my alpha helix just go in a couple of lines like this? Or does my alpha helix cross with another alpha helix? Or does my alpha helix make a different kind of structure where I have 3 alpha helixes and they are now in a triangle with each other? So this description is all tertiary structure. It is telling you how an alpha helix interacts with another alpha helix, or how an alpha helix interacts with a beta sheet, or a beta sheet interacting with another beta sheet. Now, the tertiary structure is determined by the R groups on my amino acids. So what you guys see here are all the amino acids R groups and how they can interact. And so what you guys can envision is this is one protein chain, and this is the other side to that protein chain. And so what you guys can see is they can have charged species interact, forming ionic bonds. We can have R groups that hydrogen bond. We can have R groups that form covalent bonds. So if you guys have curly hair, well, the reason you guys have curly hair is because the proteins in your hair are assembled such that these sulfurs are going to put a kink in that protein chain, giving you that curl. If you use a hair relaxer, or if you guys go out for a perm, what you guys can do is you can break this covalent bond and reset it, so now that your protein is straight versus in a curl. Other ways that my protein chain can interact is if I have nonpolar things interact with nonpolar things, so like is going to attract like. Now, this last one, there is a mistake in your book. Your book lists this as a dipole-dipole interaction, whereas OH and OH would probably be a hydrogen bond. So what I think your book wanted you to do is if you had two SH groups here. So change that to sulfur, and then you can see the last type of interaction, which is polar to polar. So long story short, I can use my R groups to get my tertiary structure. So here's the summary of all the hierarchy in a protein structure. Now, what you have to note is that each part of the amino acid dictates either the primary, secondary, tertiary, or quaternary structures. So the actual condensation, so that amide bond, is going to lead to the primary structure. This gives us our amino acid sequence. When I look at the amide bond itself, it contains a carbonyl carbon and a hydrogen on a nitrogen. Now, if I go with two different amino acids in the sequence and they're sufficiently spaced apart, this will lead to secondary structures, alpha helixes and beta sheets. Now, those alpha helixes and beta sheets are arranged in three-dimensional space, and based off the R groups, I'm going to go ahead and arrange those in space, and this gives rise to the tertiary structure. Now, the quaternary structure is something that I didn't mention, and that's when you have multiple protein chains come together. The first three are based off a single protein chain. But what happens in your body is that if you can get multiple protein chains or multiple protein polymers coming together, we go ahead and describe that as the quaternary structure. All right, Chem 1C, I hope that made sense, and remember to stay safe.